Alright, so in this video we're going to try and model the plasma membrane using little Connects models. But first, let's go over some components of the plasma membrane. So right here we're going to cover lipid rafts, which are small clusters in the plasma membrane that just have a lot of transmembrane proteins and saturated phospholipids. So if you recall from our video on lipids, unsaturated lipids are a bit more fluid, right? So here's an example of one. They have these kinks in them that allow them to spread out farther apart from one another, and they're not quite able to pack together. So this is an example of like an oil, right? So if you're dealing with unsaturated fatty acids in your phospholipids, think of oil. As compared to saturated fatty acids, which have no kinks and are allowed to pack together tightly into, I don't know, something like butter or Crisco, right? So these are able to pack together into like butter. So whenever we're talking about lipid rafts, you can sort of think of it as a chunk of butter that's floating around in a sea of oil, right? And the proteins, you can think of the proteins as potatoes. So you have potatoes and butter floating around in oil. Sometimes the potatoes are in a solid chunk of butter, which is a lipid raft. So on this diagram right here, we have unsaturated portions, and we have the lipid raft portion where we see all the proteins, some of the proteins, right? So we can just go through our list. Where can we find a transmembrane protein? Well, here's a transmembrane protein, here's a transmembrane protein, and here's a transmembrane protein. All of them are transmembrane proteins because they span the entire membrane. They come out on both ends. This would be compared to a peripheral protein, which would just be found on one side, typically the inside. So this is the outside. On the inside. Alright, cholesterol. It's kind of hard to see right here, but cholesterol is found in between the different fatty acids in the phospholipids. So right here is our cholesterol. You can upload this worksheet on the website. So let's identify the extracellular and the cytoplasmic side. So I already somewhat gave it away, but think to yourself, how could you tell the difference between the cytoplasmic and the extracellular side? The way that I would look at this is, I think of cell signaling, right? The outside of the cell needs to tell other cells what it is. So that is done through the help of these different carbohydrate bits. So the extracellular side is typically a bit more decorated than the cytoplasmic side. That isn't to say that the cytoplasmic side can't have its own quirks. It typically has the peripheral proteins that have different functions that we talked about earlier. Glycoproteins. There's two parts of this. Glyco, carbohydrate, protein is protein. So any protein that also has carbohydrates on it. This right here is a glycoprotein because it's a protein, a transmembrane protein, and it has little carbohydrates on it. Same goes for this one. It also has carbohydrates on it. So these are both glycoproteins. Glycolipids, once again, you're going to have glyco, carb, lipid, lipid. So right here is an example of a glycolipid because we have the carbohydrates attached to a phospholipid. So all of this together is a glycolipid. These are typically found in bacteria and can trigger our immune systems. Alright, so that's it for labeling this. Propose a hypothesis for the increased saturation of the phospholipids in the lipid raft region. By a quick Google search before this, I found out that lipid rafts are somewhat controversial in the cell biology community. However, we can come up with ideas of why these exist. what comes to my mind is functionality. Why not group together all the different proteins that serve a common purpose, maybe metabolism or cell signaling or whatever? Let's just group them all together into one clump of butter and call it a day. So that's what I would say, functionality. Functional proteins grouped together. You may come up with your own hypothesis, but the goal of this worksheet is to look at hypothesis and models. So moving on to models. So typically what we would do is you would take a bag of Connects toys and make your own model. You want to try and make the best membrane that you possibly can. You want to show phospholipids, transport proteins, carbohydrate chains, receptor proteins, and cholesterol. And you want to try and make it as fluid as possible and have a sidedness to it and function in transport. This is always a fun activity because it frustrates so many people, and it's supposed to show how hard it is to make a correct model, which you can't always quite do. So let's take a look at the model that I made just now. So with the toys, I made my different phospholipids like such. Right, so here they are. We have phospholipid tails and our little phospholipid heads. I even went in to throw in a carbohydrate chain. So which side would this be on? This is going to be my extracellular side because I put the carbohydrate chain here. Alright, I also made a transport protein, so I'm going to just stick that in here. It too has a sidedness to it. It has the carbohydrate chain, which shows it on the outside, and I made the proteins attaching it together on the inside a bit different. I'm going to put this wheel here to show that it's a transport protein. It's moving things from one side to the other. Could also be pumping things out. It's transporting, right? That's the wheel. I also made some little carbohydrate, or not carbohydrates, I made cholesterol. So I can smudge the cholesterol in here as such. Cholesterol tends to buffer the effects of the plasma membrane. If it's too hot, it'll keep it from becoming too liquidy and make it more solid. If it's too cold, it'll keep it from becoming too solid and keep it a bit more liquid. So cholesterol is here to help. And then finally, I made a receptor protein. My receptor protein tends to have a bit of a downfall as it doesn't span the entire membrane. I could fix that with, let's just do that, right? Now it spans the entire membrane. Why should it span the entire membrane? Because whenever our little ligand comes along and hits the receptor, it's going to have to transmit that signal throughout the membrane, right? So this too, the ligand has to be on the outside, right? Because if it's going to signal to the inside. So this is incorrect, but this is correct. So this is my model. Everybody's model is always different. Some are better than others, but each has its own strengths and its own weaknesses. I would say that a strength of this one is how I modeled the receptor and quickly came up with that. I really like the wheel to show transport, but it's not the best. My phospholipids aren't quite fluid. They kind of sit stagnant on the table, and it's not three-dimensional. That's kind of hard to do. I did get the sidedness right, and transport and receptor. So, though I got a lot of the functions of the model right, my fluidity is a bit off. Sometimes different students sacrifice other aspects of the model to make it fluid, but such is life. If you have anything like this and you can try and make a model at home, I highly recommend that you do so. Being able to do this makes for great memory formation. So good luck on making these on your own.